vou me posicionar aqui. So I think we can then start the works, and I will start thanking a lot, Henrique, for accepting our invitation and for giving your talk to us today. So Henrique Borges da Silva started as an undergraduate student at USP under the supervision of Dr. Célia Garcia at the Biosciences Institute. And in 2009, when he graduated from there, he went straight to the PhD program in immunology, also at USP, but under the advisorship of Dr. Maria Regina de Império Lima. That lasted from 2009 through 2014. And he was able to spend some time in the Gulbenkian Institute in Portugal from 2010 through 2013 under the supervision of Professor Carlos Eduardo Tadocoro, sorry. And after that, he came back. He graduated in 2004 and 2014 stayed for a little postdoc and went back. Now I went to the U.S. to the University of Minnesota and stayed as a postdoc from 2015 through 2020 at the University of Minnesota Twin Cities under the supervision of Dr. Stephen Jameson. Was a very productive time over there. He started working with T cells and the role of ATP in regulating their function. In 2019, he was awarded the K99 ROO Career Development Grant from NIH and started as an assistant professor at the Mayo Clinic in Phoenix, Arizona in 2020. And I thank again Maria Bello for making this bridge between us and intermediating the invitation for Henrique to talk to us. So Henrique, thank you again, and the floor is yours.
All right, thank you so much. Sorry about that. Like one of my screens is like having an issue connecting. So let me share my screen again, and then we can start. Let me just do something to avoid the issue in the next few minutes. Of course, the technical issues would start as soon as we start the seminar, right? But we'll be fine. Don't worry. You guys can see my screen now, right? Let me just swap it to enter mode. There you go. Now we're talking.
Perfect. Thank you.
Yeah, no problem. Thank you so much for the invitation. It's always a pleasure to talk to the people at Brazil about the research that we do here at Mayo Clinic in the nice and cool Phoenix, Arizona. It's not nice and cool; it's hot as hell outside. As Miriam mentioned, most of our research focuses on sensing of extracellular ATP and how can that regulate T cell responses. I'm going to start by saying an important disclaimer that I mean, I don't really do the job. I just lucky enough to have a really good group of trainees and postdocs in my laboratory. This is our laboratory. We need an updated picture. They're the ones that really do the research. So I always start with the acknowledgments of the group. And today we're going to see some of the research done by some of them. As the name of our laboratory, which is Laboratory Immunity to Danger Signals, or LIDS, say, we study danger signals and how can they regulate immune responses. I think some of you are probably know what danger signals are. They're basically signals from self that are associated with situations what we call danger to the host. So if you have an infection, or if you have a tumor growing in your body, or if you, I don't know, burn your foot in a George Foreman grill like Michael Scott on The Office, you're going to have any kind of injury and cell death. You're going to have the release of a lot of these self molecules that not necessarily they're going to act as self-peptides. That's a different thing. That's autoimmune responses. But they're going to have certain molecular patterns that can be recognized by specialized receptors. So that's very similar in a way to what happens with toll-like receptors or any other pattern recognition receptors. The only difference is that the patterns that we recognize on PRRs are derived from pathogens. And these patterns are they're recognized, they're derived from self molecules, but those self molecules are not normally present in the outside of the cell. And of course, the idea that these danger signals can regulate immune responses was postulated, or among the first to postulate was Polly Metzinger, all the way back in the 90s. And that's her with her dog. That's a famous story with her dog in a GXMed paper. We can talk more about that later. And as I mentioned before, danger signals that can directly regulate immune responses, both innate and adaptive immune responses, because they're both myeloid cells and lymphocytes that can express receptors for things like, of course, extracellular ATP, like we're going to talk today, IL-33 as well, which is, although it's a cytokine, it is released pretty much as a danger signal, at least its active form among others. And our laboratory, but we do have a couple projects focusing on IL-33. A lot of it is a collaboration with Dr. Denise Fonseca, that's at the University of São Paulo. But today we're going to talk about ATP, which ironically is not what I worked on during my PhD years on the Regina's lab, but everybody else on Regina's lab was working with extracellular ATP. So that's where I got the idea. And the idea of our laboratory is, as I mentioned, is how the danger signals regulate immune responses. And again, as I mentioned before, extracellular ATP is an excellent example of a danger signal. In many circumstances, you're going to have the accumulation. Let me put the laser pointer to make life easier, of ATP in the extracellular environments. That can be done. That can be due to cell damage. Of course, if you have overwhelming cell death, like what occurs in the middle of a tumor, you're going to have a lot of ATP being spit out to the extracellular environment. Nucleotide transporters such as pannexin-1 can also be important on that. We also investigate the role of pannexin-1 on T cell responses. There's also possibility for vesicular exocytosis. But the important thing for the talk today is that once in the extracellular environments, extracellular ATP can bind to these receptors, which a lot of you guys probably already know from the works from Regina and Hobson at Rio de Janeiro, called purinergic receptors. More specifically, we're talking about here P2X receptors. Those are ion channels that bind to extracellular ATP. To our knowledge, they exclusively bind to extracellular ATP. And then you have, well, of course, intracellular calcium influx or sodium flux. And there are 7 known P2X receptors, at least in mammals. And they mostly do similar jobs. I mean, like they are ion channels that leads to calcium influx. And they also can inducing sodium influx. And in the case of this particular ion channel called P2RX7, you can also have potassium efflux. And that's not the only reason why P2X7 is what we call a different receptor. It also has a low affinity compared to other P2X receptors for extracellular ATP. That means that you need to have relatively high amounts of extracellular ATP to activate this receptor. That's why traditionally P2RX7 has been considered a bona fide danger signal receptor. It is not exclusively expressed by immune cells, but it's preferentially expressed by immune cells. It also has low to no desensitization. So most P2X receptors, as most ion channels, of course, once the ligand binds to it and you have calcium influx in that case, there's a quick desensitization of the receptor, meaning that there's a period of time where that particular receptor is not going to be active. That's not true for P2X7. There's changes in the structure of P2X7 that make this receptor not able to desensitize. And there's also quite high level of polymorphism of P2X7, even among healthy individuals. That there are even some studies in the past that show close to 10% of heterozygous loss of function SNPs in P2X7. So, especially for immune responses, we always thought that P2X7 could play an important role, and not only us, but many others, right? There's a classic role attributed to P2X7, which is activation of the NLRP3 inflammasome in innate immune cells. But the other thing that it's important to note is that function of P2X7 could impact immune responses pretty much everywhere in the organism, right? When we talk about immune responses, usually we kind of focus on uh, what's going to happen in the circumstances in the spleen or in the lymph nodes or in the circulation. But it's highly likely that you're going to have impacts of extracellular ATP and P2X7 organism wide. And the two main reasons for it is that in any tissue, danger signals can be released because you can have injury or infection or tumors growing virtually anywhere. And it is, of course, more and more acknowledged that immune cells, not only macrophages, but also T cells, can be found virtually anywhere in our body, especially with more ancient exposure. And the focus of the presentation today, and it's a large focus of our laboratory, is on tissue-resident immune responses or tissue-resident T cell responses. And this is just a cartoon that I'm not going to enter too much detail, but it's just to illustrate that in different tissues, you're going to have a whole bunch of different types of immune cells. And this is going to be crucial to provide quick responses in these barrier tissues. And in the case of tissue-resident memory T cells, that's going to induce protective responses against the reinfection in this tissue. So if you think about when you have an infection in the lung, such as influenza infection, you are going to induce the formation of tissue-resident memory T cells in the lung tissue, both in the epithelium and in the parenchyma. And that's going to be very important to provide a quick an efficient response against a reinfection with the same pathogen, or at least with a pathogen of the same type, right? Uh, and this is the basis of the function or the biological relevance of tissue-resident memory T cells. And that is something that, again, I could spend hours talking about how to divide memory T cell responses. There was a whole debate in previous Keystone meetings on memory T cells, on how to divide memory T cells. You can divide them based on function. You can divide them based on longevity versus lack of longevity, but you can also divide them based on migratory patterns. And then, as I mentioned before, this is going to be our focus today. You have tissue-resident memory cells, which main function is to survey and reside long-term in these barrier tissues. You can find tissue-resident memory cells in the skin, the gut, lung, brain, liver, female reproductive tracts, etc. And of course, the counterpart, you have circulating memory cells, which can also be subdivided into central memory cells, which are enabled via by rule to go to non-lymphoid tissues and effector memory cells to survey pretty much everywhere. But today our focus are going to be on tissue-resident memory cells. And there are many markers that we can use to define or to more or less define tissue-resident memory cells. None of them are perfect because there's, of course, heterogeneity among tissue-resident memory cells. But the important thing to remember about TRMs, that's what I want to call them, is that typically they're going to express high levels of molecules that are going to help in their retention into tissues. And in counterpart, they're going to express low levels of molecules that are going to help in their egress from the tissue. So every time you think about markers of retention, such as CD69 and CD103, those are going to be what we call TRM markers. And in counterpart, you have markers of egress, such as L-selectin or KLRG1, or the expression of this transcription regulator called KLF2. Those are going to be associated with the cells getting out of the tissue. And this is old hits, right? When I was still a humble postdoc at Steve's lab in Minnesota, to show that we and others found that memory T cell subsets, CD8 T cell memory subsets, can express actually quite high levels of P2X7, especially tissue-resident memory cells in places like the small intestine, intraepithelial site. So a lot of, especially epithelial TRMs, can express quite high levels of P2X7. This is something that we found almost now 10 years ago, actually. My God, it's been 10 years since I started my postdoc at Steve's lab. I'm getting old. And of course, many other researchers on the TRM field have found that. And as a matter of fact, nowadays, P2X7 is considered a marker or a quite faithful marker, at least in mouse models, of epithelial tissue resident memory cells. So this is one of the first observations that I made when I was a postdoc at Steve's lab. So that kind of made us curious to understand what could be the biological significance of P2X7 for tissue resident memory cells. And that's important because initially we thought that by knocking out P2X7, we will have increased accumulation of tissue resident memory cells because in many circumstances for T cells, and a classical example for that is follicular helper CD4 T cells, expression of this extracellular ATP receptor is deleterious for their survival. So usually, whenever you have a knockout or a blockade of this receptor, you tend to have accumulation of T cells, and this is partly due to this unique characteristic of the P2X7 receptor, which is the induction of apoptosis when you have way too much ATP. So we did what any immunologist would do: would block and knock out P2X7 on T cells and study the numbers and the phenotype of memory cells in multiple tissues, right? So to study CD8 T cell memory, and more specifically tissue resident memory, we use this very common model that to study virus-specific T cells infection with LCMV Armstrong. It's a systemic virus that induces acute infection, so the virus is controlled quite quickly in a week, and then you can study using multiple systems, tetramer staining, or transfer of these CD8 T cells called P14 CD8 T cells, which have antigen specificity to the LCMV Armstrong. You can study the ability of virus-specific T cells to form memory in distinct tissues. So what we did, among many other experiments, of course, is what we call co-adoptive transfer of P14 cells, where we transfer a one-to-one mix of wild type P14 T cells and P2X7 knockout P14 T cells into mice infected with LCMV. And then we tracked the formation of memory cells over time in multiple tissues. I mean, every single tissue you could imagine we took from the mouse. It's quite gruesome experiment. And this is just one piece of data showing that although the initial generation of memory cells in the small intestine intraepithelial compartment doesn't seem to rely on P2X7, at least in numbers. The phenotype is different. We're going to talk about that later. Over time, their survival is affected in the absence of P2X7. And this is true in many other tissues: female reproductive tract, male reproductive tract, brain, salivary gland, kidney, you name it. So pretty much everywhere we can have tissue resident memory cells, you need expression of P2X7 for these tissue resident memory cells to establish. And as I mentioned before, we use many other experimental strategies of tracking tetramer-specific endogenous T cells, using CRISPR-Cas9 to knock out P2X7, using pharmacological inhibition, etc. These are old hits. I mean, I was still a postdoc in Steve's lab. Of course, now I moved on and became my own lab. And one of the things that we're trying to understand in our laboratory is how P2X7 can regulate the longevity of tissue resident memory cells and the survival of these cells. And this cartoon, I'm not going to enter in too much detail, but it basically establishes the different subsets of memory cells that can form over time in response to, in this example, a virus. As you can see, you can have multiple subsets of memory cells that all arise from memory precursors. So during the effector stage of immune response, these CD8 T cells that clonally expand, they can become heterogeneous in function and in fate. In a way that you have a large population of terminal effectors, which are going to perform effector function. They're going to eliminate the virus. Most of them are going to die. Some of them are going to become this kind of circulatory memory cells that we're not going to talk about today. And then you have this population of memory precursor cells that can give rise to or bona fide memory populations that can be both in the periphery or in lymphoid tissue. And of course, it is possible that the importance of P2X7 for tissue resident memory longevity per se could be imprinted early on. P2X7 is upregulated more or less early after an infection or antigen exposure. So it is possible that what we can be seeing here is that the expression of P2X7 is promoting the development of what we call a long-lived TRM precursor. That is a possibility. Another possibility, and that's not completely excluded from the other one, is that you need simultaneously in multiple other tissues extracellular ATP signals to promote their long-term survival, per se. So we wanted to explore these two possibilities, and I was still actually a postdoc when I did that by using a tamoxifen-inducible knockout system. So basically, with this system using tamoxifen treatment, you can induce the knockout of P2X7 at different time points after infection. And in these experiments, basically, you have tamoxifen-inducible knockouts, P14 cells and wild-type P14 cells, in this case, control mice, cells, that we co-adoptively transfer again into mice infected with LCMV. And in some experiments, we treated these mice with tamoxifen after memory cells were already formed. So in this case, now we're inducing the ablation of P2X7 in these cells after they already formed memory populations. And what we can see here is this is a ratio between this inducible knockout and the wild type cells, is that in multiple tissues, you have a decay in numbers of these inducible knockout P14 cells. So these results strongly suggest to us that at least in part, you need expression of P2X7 to remain high on this tissue resident memory cells for them to survive long term. So it is highly likely that these cells, they continue to sense extracellular ATP over time. It doesn't need to be constantly, but at least over time to sustain their long-term survival. However, with this tamoxifen system, we can also knock out P2X7 at the early stages of infection, kind of like mimicking what we find with our germline knockout system. And now we find, to some extent, similar results. I mean, you also need expression of P2X7 at that effector stage of immune response, which is before these cells are going to the tissue to induce this ability of these cells to survive for a long term. So it looks like you need expression of P2X7 at both stages, which is great. I mean, like, it's important to know that. But the first question that our laboratory, now we're going to enter into Borges da Silva laboratory years, that we wanted to understand, okay, but how does P2X7 expression is promoting the longevity of tissue resident memory cells? I mean, with those experiments, we kind of answer when, right? But we don't know how. And we were especially intrigued by these effects early on. This possibility that you need the expression of P2X7 and probably their ion channel function to induce this early imprinting for longevity. And we started looking back. Instead of looking at the phenotype of the memory cells, we started looking at the phenotype of the effector cells in the spleen at an early stage after infection. And what we found is that there is already, to some extent, upregulation of P2X7. It's not too high of an expression, but you can kind of discern cells that express more P2X7 than others. And if you divide the cells arbitrarily into P2X7, quote unquote, high or low, you start seeing already some patterns in which these cells that are expressing some P2X7, they're already expressing some markers that are associated with tissue residency in memory. I'm not showing here, of course. And in counterpart, these cells, they tend to express lower levels, these high P2X7 cells, lower levels of markers associated with either tissue egress or less survival, such as KLRG1. So it looks like there's already some kind of functional heterogeneity appearing early on during the effector stage of immune response that could be marking their ability to become memory cells or not. So to actually test that, we did secondary adoptive transfer experiments where basically what we did is we adoptively transfer P14 cells. Now it's only wild type P14 cells into mice that are then infected with LCMV Armstrong. And then after 4.5 days after infection, so at this point, there are no T cells in tissues. These T cells are all in the spleen. They're still clonally expand. We sorted these P14 cells based on P2X7 expression. So basically, we selected the top 20% and the bottom 20%. And then we secondarily adoptively transferred these cells into infection-matched mice. So in this experiment, basically what we're going to be testing is the fate of this P2X7 high versus low cells, and to see if one cell versus the other, which one is better into forming memory populations in multiple tissues. And we're showing here just an example for small intestine IL, but it's the same for other tissues. Both the initial establishment, so if you look 15 days after the transfer, as well as their survival long term, so this is a 90 days after the transfer, uh, it's better if these early effector cells are expressing higher levels of P2X7. So when you have these early effector cells already expressing higher levels of P2X7, that seems to mark these cells to become better at forming tissue-resident memory cells. And not only that, but they're better at forming tissue-resident memory cells that survive longer. So this is additional evidence that you need expression, which is unequal, of this extracellular ATP sensor to imprint, to some extent, an ability of these cells to become long-lived TRM cells. So, of course, we wanted to understand molecularly how this is happening, and of course, we're still doing that. And what do you do when you don't understand for sure what something is happening molecularly? You do whole genome transcriptomics, and that's what we did. We did bulk RNA-seq comparing wild type versus P2X7 knockout early effectors, or P2X7 high versus low early effectors. And here I'm just showing an example of the heat map showing the top differential expressed genes between wild type and knockout cells. But it's a very similar scenario for high versus low. And this is a list of genes that we see that are commonly high on either wild type or P2X7 high early effector cells. A lot of them are associated with long-lived memory populations, and some of them are associated with tissue residency. And in counterpart, there are a lot of genes associated with tissue egress or short-lived memory populations that are expressed at higher levels on P2X7 low or deficient early effectors. And one particular transcriptional regulator caught our attention. It's called ZEP2. So ZEP2 is a transcriptional repressor that is necessary for the formation of short-lived effector cells. What that means is that if you have expression of ZEP2 that downregulates the expression of multiple molecules that are necessary for the formation of memory populations. Not much was known about the effect of that on tissue-resident memory cells. So we wanted to understand what could be the connection between P2X7 and ZEP2. So for that, we used the same adoptive transfer system that we used before. You have wild type or you have P2X7 knockout P14 cells. And now instead of simply just transferring these cells into infected mice, we also use CRISPR-Cas9 to knock out ZEP2. CD4 here is a negative control CRISPR guide. So now we have 4 experimental groups. We have wild-type CD8 T cells. We have wild-type CD8 T cells that were knocked out of ZEP2. And then we have the same, but for P2X7 knockout. So de facto, we generated P2X7 double knockout with ZEP2. And all we can see here from the data on the right is that the knockout of ZEP2 rescued the ability of P2X7 knockout CD8 T cells to form tissue-resident memory populations. So what that shows to us, or that indicates to us, is that you need the expression of P2X7 to induce, to some extent, the repression of ZEP2. And that is partly the reason why you need P2X7 expression for tissue-resident memory cells to form. Now, as I mentioned before, ZEP2 is a master transcriptional repressor. So we were a little curious to see whether this was the opposite, right? Whether instead of having P2X7 necessary for ZEP2 downregulation, you also, what you need is actually something else to downregulate ZEP2, and that is what driving P2X7. So we wanted to understand what is upstream of what. So it looks like ZEP2 doesn't necessarily repress P2X7, because when we look in our ZEP2 knockouts, CD8 T cells, they still have similar levels of P2X7. So that kind of suggested to us that it is highly likely that what's happening is that you have P2X7 and extracellular ATP sensing occurring upstream of ZEP2. And one of the things that we know can repress ZEP2 is TGF beta signaling via TGF beta receptor 2, which is another signal crucial for the formation of tissue-resident memory cells. So we wanted to test whether the expression of ZEP2, which indeed is aberrantly high on P2X7 knockout CD8 T cells, can be repressed if you induce expression of TGF-beta receptor 2. And indeed, when we overexpress TGF-beta receptor 2 on P2X7 knockout cells, you induce a repression of ZEP2 in these P2X7 knockout CD8 cells, which is correlated with our better ability of these cells to form memory populations in tissue. So this is from a previous work that I did when I was, again, a postdoc. When we overexpress TGF-beta receptor 2 on P2X7 knockout cells, we indeed rescue the ability of these P2X7 knockout cells to form tissue-resident memory populations in the gut. So all this together show that you need P2X7 to induce TGF-beta receptor 2 expression. We have other data showing that. That's necessary to induce the tissue-resident memory subset in different tissues and their longevity, in part through the repression of ZEP2. And then finally, we also found that this upregulation of tissue-resident memory markers that's dependent on P2X7 is probably occurring through calcium influx. This is an important question. I mean, the canonical function of P2X7 is calcium influx, but it's not the only function of P2X7. So by using a calcineurin inhibitor called FK506, or stacrolimus actually, we can repress to some extent the upregulation of a tissue-resident memory marker called CD103 in wild-type cells, but no effect is observed on P2X7 knockout cells. So this leads us to this working model. It is not a complete working model, of course, which suggests to us that P2X7, probably through calcium-mediated pathways and calcineurin, is inducing the upregulation of TGF-beta receptor 2. That is repressing ZEP2, and that's important to drive the TRM long-term establishment program, because ZEP2 typically blocks that. Now, right now, what we're doing is that we're trying to further advance our understanding of this pathway, because we understand that there are pieces of the puzzle missing here. And we looked back in our RNA-seq data, and we found an interesting dichotomy in expression of ID3 and BLIMP1. So ID3 we found to be highly expressed by effector cells that have high expression of P2X7. And BLIMP1 is highly expressed on early effector CD8 cells that lack expression of P2X7. Why are we interested in that? And that's because of this work from Nana Goldrath's group a few years ago, showing that you can use ID3 and BLIMP1 expression to resolve the heterogeneity of tissue-resident memory cells. So we love to put things into boxes, the people that do T-cell research, right? So that's one other chapter of that. So basically what they found is that among the cells that initially seed tissues, such as the guts, you're going to have tissue-resident memory cells that express very high levels of ID3. And those cells tend to be longer lived. They tend to survive for longer. They tend to express higher levels of molecules associated with memory, such as TCF1 included. On the counterpart, BLIMP1 expression, which historically was actually proposed to be important for TRM formation, it does induce TRM formation, but it induces a large population of TRM cells that actually are not going to be long-lived. So these cells, they tend to be shorter lived. They tend to express lower levels of memory associated markers. So basically as a summary, what you're going to have is the formation of two major clusters of tissue-resident memory cells, and they likely have complementary functions. But one of them is longer lived and one of them is shorter lived. So our question now, based on our RNA-seq data, is that does P2X7 expression, that unequal expression that we're seeing at the effector stage of the immune response, is these marking cells that are going to form heterogeneous responses, or is P2X7 dictating to some extent the heterogeneity of tissue-resident memory cells? And this is work that is ongoing. We're not done yet. Some of it is being done by Caio Salgado, who's a very talented postdoc in our laboratory. He was a grad student at USP. Some of them was done by Chloe Leff, another very talented undergrad in my laboratory. Now she's at UPenn as a grad student. We started by looking at the expression of BLIMP1 and tracing correlations with the expression of P2X7 in our tissue-resident memory cells. And not surprisingly, we see populations that express high levels of BLIMP1, populations that express low levels of BLIMP1, and the ones that expresses higher level of BLIMP1, they're the ones that tend to express the highest levels of P2X7. The other ones, they still express P2X7, but it's at lower levels. So there's to some extent an inverse correlation between at a protein level between BLIMP1 expression and P2X7. So to some extent that agrees with our RNA-seq data. So their next step is to understand, okay, what's going to happen with these subsets of tissue-resident memory cells, this BLIMP1 versus ID3, if you knock out P2X7? And what we see is that, of course, numerically, there are more wild-type than P2X7 knockout cells. Now we're looking at endogenous tetramer-specific cells. And these mice, they are transgenic for both ID3 and BLIMP1. Those are difficult molecules to stain by flow cytometry. In a way that you have ID3 GFP versus BLIMP1 IFP. And when we look at 2 months after infection, so at this point, a lot of the short-lived tissue-resident memory cells, they already kind of died off. The vast majority of your tissue-resident memory cells in the guts, they are going to be ID3 positive. You're going to have very, very few BLIMP1 positive cells. In this example, it was 0, but like in reality, you have very few BLIMP1 positive cells. So initially, we expected to see less ID3 expression and more BLIMP1 expression, right? So, but when you look at simply ID3 expression, you still see expression of ID3. I mean, it's a little less, but it's not significant difference. So the absence of P2X7 is not blocking the expression of ID3 by these tissue-resident memory cells. Instead, what it's doing is that it is impeding the downregulation of BLIMP1, at least at a population level. You still have a lot of expression of BLIMP1. You do have a lot of cells that only express BLIMP1. And you start having these cells that co-express BLIMP1 and ID3. So that suggests to us that these tissue-resident memory cells, they're unable to downregulate BLIMP1. And again, this is just one example, but if you look at absolute numbers, at this point, you have like 5 times less cells on the P2X7 knockout. So what this shows to us is that you need the expression of P2X7 for these tissue-resident memory cells to downregulate BLIMP1, even the ones that are upregulating ID3. To further understand how could that be transcriptionally, how could that be occurring transcriptionally, we sorted what we call TRM precursors. So this is at 1 week after infection, based on BLIMP1 expression. So basically what we sorted is wild type IEL cells that are BLIMP1 positive versus BLIMP1 negative that express or not P2X7. And then we performed RNA-seq. And this is a PCA plot. And what this shows is that a lot of the differences are, at that point, transcriptionally dictated by P2X7. So the vast majority of the PC1 effects are dictated by P2X7. But what you can see here is that the wild-type cells, regardless of BLIMP1 expression, they cluster kind of together. But there's already an aberrant expression at a transcriptional level in the absence of P2X7, even among the ones that don't express BLIMP1, and even more the ones that express high levels of BLIMP1. We're still analyzing this data. We actually got this RNA-seq data 2 weeks ago, so I'm still trying to make a sense of it, but there are a few patterns that are starting to appear. Don't worry, I'm not going to force you guys to read every single one of the lines here, but when we start seeing the differentially expressed genes between the populations here, so here, this is wild-type BLIMP1 negative. This is wild-type BLIMP1 positive. This is BLIMP1 negative on the knockout, and this is BLIMP1 positive on the knockout. So you don't need to memorize this. But what is important to notice is that, especially on the BLIMP1 negative P2X7 knockout TRM precursors, but also for the BLIMP1 positive, you have higher expression of dysfunction-associated genes or exhaustion-associated genes, such as LAG3 and CTLA4. There are a lot of tissue egress genes, so genes that are usually associated with cells that are not going to become tissue-resident memory cells that tend to be highly expressed on the P2X7 knockout IL precursors. And then in that case, it's especially the ones that are BLIMP1 positive, such as CX3CR1 and L-selectin. And I think this is, to me, the most important cluster of genes. So those are genes that are associated with long-lived tissue-resident memory cells that were found by the Goldratt group a few years ago. So TCF7, XCL1, NSG2, KIT, SLAMF6, L7 receptor, etc. So predictably, they're higher on BLIMP1 negative wild type TRM precursors compared to the BLIMP1 positive. So that would suggest that these cells are indeed the long-lived TRM precursors. Now, if you look at this gene expression level in the P2X7 knockout cells, the ones that are BLIMP1 negative at that point, they still express those genes. But the ones that are BLIMP1 positive also do. So what that suggests to us is that the absence of P2X7 blurred the lines between the long-lived TRM precursors and the short-lived TRM precursors. And together with the expression of dysfunction-associated genes and tissue egress genes that could suggest that indeed these P2X7 deficient cells, even if they initially have this gene signature of a long-lived precursor, they are perhaps marked not to survive that well or not to stay that well in that tissue. Of course, these are things that we still need to investigate, right? But what that suggests to us, what the data that we have so far suggests to us, is that in the presence of P2X7, you need expression of calcium to induce TGF-beta that represses AB2, that's important for the TRM establishment at the same time, and this may or may not be correlated. You need P2X7 expression to repress BLIMP1, which is also going to drive this long-lived TRM establishment. And there are many other pieces of the puzzle that we're currently investigating, such as the involvement of BCL6 as a very famous downregulator of BLIMP1, the involvement of other regulators, and whether dysfunction or egress are the ones that drive the P2X7 knockouts, absence of long-lived memory cells. So in the final 15-ish minutes or 10 minutes, I'm going to move to CD4 T cells and talk a little bit about another work that we've been doing here in our laboratory. I'm going to try to be fast. And it is important to note that you don't only have CD8 T cells forming tissue resident memory populations, but also CD4 T cells. And especially our focus now is on respiratory immunity. The presence of CD4 tissue resident memory cells in the lungs is very important to sustain protective immune responses against antigens or pathogens that are airborne, such as influenza. And influenza, of course, remains a significant public health concern. It causes a few 100,000 deaths globally per year. In the United States alone, there's over 40 million cases annually. And last year, we actually had a very, very strong flu season here in the United States. And there is a gold rush. I mean, it's kind of a forbidden word now here in the United States for universal influenza vaccines. Well, there are a lot of different types of influenza vaccines. A lot of them are commercially available. Most of them are what we call subunit vaccines, and they do provide protection against the seasonal influenza, but they don't provide what we call universal protection against influenza. And part of it is due to the characteristics of the influenza virus of high levels of mutation, antigenic shift and drift, if you remember your classes. And what is believed about a potential universal influenza vaccine is that you need ways to induce long-lived responses that are tissue resident, both tissue resident B and T cells. And when we look at influenza-specific CD4 tissue resident memory cells, they are a heterogeneous population, not surprisingly. This was actually discovered by or unveiled by back-to-back publications by Ji Sun from University of Virginia, one of our collaborators, and Carolyn King from University of Basel. And what they found is that in response to influenza infection, you tend to have two major clusters or populations of CD4 tissue resident memory cells. One that expresses very high levels of Th1 associated markers such as TBAT and PSGL1, and one that expresses pretty high levels of TFH associated markers such as BCL6. Those that are TFH-like, they coined them tissue resident helper cells or TRH cells. And these TRH cells, they are believed to be quite important for the amplification of other lung immune responses. So these TFH-like tissue resident helper cells, they seem to perform in the lung tissue functions that are to some extent very similar to what the TFH cells will be promoting in lymph nodes, for example, which is the amplification of B cell responses and CD8 T cell responses, which is a non-canonical function of TFH cells. So This is actually a work that another very talented postdoc in our laboratory, Igor Santiago Carvalho, has been doing. We're trying to understand where does P2X7 fit in this story. And the first thing that he observed is that, and of course others observed as well, is that in response to influenza infection, we use a lab-adapted influenza infection expressing this peptide called 2W that allows us to look at antigen-specific responses using distinct MHC class 2 tetramers. We observe three actually major clusters of tissue-resident memory CD4 T cells in the lungs. One that is very similar to that TRM1 population, one that is an intermediate population, which actually has been more and more annotated in the literature, and what we call the bona fide TRH population, which expresses very high levels of a lot of TFH-like markers. And what we found is that these TRH cells are the ones that express the highest levels of P2X7. They actually express very high levels of P2RX7. At a first glimpse, this is not surprising because follicular helper cells also express very high levels of P2X7. And again, we initially expected that TFH cells, the TRH cells, would actually be increased in numbers when you knock out P2X7, because this is what we find for TFH cells, right? As I mentioned before, in many cases, right? That's what Igor tested. He used wild type and T cell specific P2X7 knockout CD4 CRE, P2X7 flock mice. He infected these mice with the lab adapted influenza, and he tracked the numbers and percentages of the different subsets of tissue-resident memory CD4 T cells. And what we found, again, contrary to the initial notion, is that you need expression of P2X7 for, or largely need the expression of P2X7 for these tissue-resident helper cells to form. And percentage-wise, you have an increased accumulation of these TRM1 cells. And this is in contrast to what we find in the mediastinal lymph nodes. So actually, in this case, we don't find a difference between wild type and T cell specific P2X7 knockout cells. And this is a T cell intrinsic phenomenon. So instead of using a co-adoptive transfer of transgenic TCR T cells, we used another system. It's a mixed bone marrow chimera. So basically, we literally irradiate mice and reconstitute them with a one-to-one mix of bone marrow cells from wild type or T cell specific knockout mice. Waited 2 months for reconstitution of the system. So now you have a one-to-one chimera mix. Infected these mice, and what we see is that in the same mice, now in an intrinsic way, there's still a reduction in percentage and also in numbers of these tissue-resident helper cells in the absence of P2X7. So you need expression of this extracellular ATP sensor for the formation of these tissue-resident helper cells. We also tested whether you need expression of P2X7 for the survival of this tissue-resident helper cells. So now, instead of using a tamoxifen-inducible system, which we're still establishing in our lab, we use treatment, pharmacological treatment, inhibition with P2X7 inhibitor called A438079. And we treated these mice intranasally, if I remember well. Igor can correct me if I'm wrong. After the formation of the tissue-resident memory cells in the lungs. And what we found is that, again, you need the expression of P2X7 for the survival of these tissue-resident helper cells. In a way that if you block P2X7 after the formation of the tissue-resident helper cells, we also see this phenotype. And as a matter of fact, we also did intraperitoneal treatments with this inhibitor, and we see the same results. We're now trying to understand mechanistically what can be happening. I mean, like, is this pretty much the same mechanism as we're seeing for the CD8 T cells? And we deem that unlikely because CD4 TRMs tend to be quite different in phenotype and localization compared to CD8 TRMs. So for that, we to understand that, we did single-cell RNA-seq analysis of these flu-specific lung CD4 T cells. Not surprisingly, we found 3 major clusters of genes or gene expression cells. One that resembles Tregs, one that resembles TRH cells, and one that resembles TRM1 cells. And when we compare wild-type and P2X7 knockout cells in these TRH clusters, we found a few interesting things. So the first thing is that we found higher expression on the wild-type TRHs of several mitochondrial genes, a lot of genes associated with survival, such as BCL2, and this gene called CD55. We're going to talk about that in a slide, a couple of slides later. And when we look at pathways that are enriched on the wild-type TRH cells, we find electron transport chain and protein biosynthesis. And usually when we find electron transport chain pathway enriched or mitochondrial genes enriched, that usually means that the amount of mitochondria in that particular cell is different. So we track that using MitoTracker green staining. And what that data suggests to us is that the fewer P2X7 knockout TRH cells, they do indeed have lower mitochondrial mass or mitochondrial content inside of their cells. At the same time, we used CNIT analysis, which allows us to look via puromycin incorporation, the dependence on either mitochondrial metabolism or glycolysis for protein synthesis. And what we see is that the P2X7 knockout TRH cells, although they have less mitochondrial content, they depend more on their mitochondria for protein synthesis. What means is that they have lower capacity to use glycolysis or aerobic glycolysis for protein synthesis. So right now we're operating under the hypothesis that what's happening here is that in the absence of P2X7, there is a decrease in the ability of these TRH cells to use aerobic glycolysis for their metabolism. That means that they need to use mitochondrial function. But at the same time, that leads to a progressive loss of mitochondria in these cells by phenotypes, by mechanisms that we're still not sure. So as a net result, the overall metabolic capacity of these cells is going to be decreased, and that's going to contribute to their lack of survival in the tissue. And we also, agreeing with this notion, we use ACAR treatment. This is a drug that activates the MPK pathway and increases mitochondrial biogenesis to see if we could rescue the ability of TRH formation in the absence of P2X7. And what we see is that indeed P2X7 deficient mice that were treated with this ACAR, they tend to have higher percentages or to some extent a rescue in the formation of these tissue resident helper cells. So that is another result going in the direction of the necessity of P2X7 expression for the establishment and the maintenance of this mitochondrial content on these cells. And of course, we're still evaluating that. Another molecule that we did observe to be different between wild type and knockout cells is CD55. And that's something that I was terrified when I saw that is a molecule associated with the complement system, which, with the exception of a few people like Lourdes at USP, everybody is terrified to work with, and is actually DAF, or is an inhibitor of the membrane attack complex. And we actually were able to confirm that as a severely downregulated on P2X7 knockout tissue resident helper cells. And what we are observing now is that CD55 can actually act as a co-stimulatory molecule. So T cells in other contexts can bind to CD97 that can be expressed by either B cells and dendritic cells and other immune cells, and that can drive a co-stimulatory signal to these cells. So we're still evaluating that by several different experiments, but we are wondering whether the presence of CD55 on these cells can help in an additional co-stimulatory signal to drive this mitochondrial biogenesis and cell survival. But this is part of the puzzle we haven't solved yet. And then finally, in the last minute or so, we got to put too much stuff on this presentation. We wanted to understand, okay, what will be the impact of P2X7 for the function of these tissue resident helper cells? And as I mentioned before, the function that's classically attributed to these cells is to help CD8 T cells and to help B cells. We wanted to understand especially what's happening to B cells. And for that, we use an homologous challenge system. So basically, we infected mice with a low dose influenza and then challenged them with a lethal dose of influenza. And what we observe is that the T cell specific P2X7 knockout cells, they have significantly higher levels of viral titers in the lungs after a few days of challenge. What this suggests is that in the absence of P2X7 in these T cells and in the absence of TRH cells, or the loss of TRH cells, these mice are more susceptible to a challenge. They're not able to control the virus very well. This is associated with more inflammation in the tissue, which makes sense, where you have more virus staying. And this is associated also with having more TRH cells now expanding in response to the challenge. And which is not observed to the same extent of the P2X7 knockout. As I mentioned before, a homologous challenge is mostly related to a B cell response. So we wanted to evaluate the B cell response. So the first thing that we wanted to understand is the numbers of B cells after challenge. And that's the part where we got a little confused at first, because we actually saw more B cells in the P2X7 knockout mice after challenge. So we were a little confused about that. So why do we have more B cells? Are these B cells equally functional? So the one thing that we wanted to understand is, okay, where are these B cells? So we did immunofluorescence of the lungs. Igor did immunofluorescence of the lungs. And curiously speaking, we didn't see a lot of aggregates of B cells and CD4 T cells after challenge, which is usually what you see when you form these memory populations without challenge. Instead, what you see is that a lot of B cells that are kind of intercalating with CD4 T cells. So we haven't quantified that yet, but there is a lot of potential contacts between CD4 and B cells here, suggesting that there's a lot of B CD4 T cell interactions in this tissue, but not a lot of IBALT or IBALT-like generation. In contrast, what we see in our P2X7 deficient mice is that it looks like you do have more B cells, but these B cells, they don't seem to be in contact with the CD4 T cells. So the CD4 T cells in the P2X7 knockout, they seem to be excluded from these areas that are rich in B cells. So that usually suggests that you're not going to have necessarily these B cells responding specifically to the antigen. So that suggests that a lot of these B cells might be bystander B cells. So thanks to Gabriel Victoria, we got the AJ tetramer to mark specific B cell responses. And basically what we see is that we almost don't have flu-specific B cell responses occurring in these mice. So what this suggests to us is that in the absence of these TRH cells, or in the decay of these TRH cells in the P2X7 knockout mice, you are not being able to generate the proper antigen-specific B cell responses. Instead, what you have is an invasion of a bunch of bystander B cell responses, which might be correlated with the inflammation and the viral titer that's still present in these mice. And as a consequence, when we compare the amount of antibodies in these mice, what we see is that in response to challenge, this T cell specific P2X7 knockout mice, they are unable to produce flu-specific antibodies. So that suggests that the absence of TRH cells, in the absence of P2X7, you have lower ability to specifically form the antigen-specific responses that you need to control this virus challenge. Instead, what you have is an accumulation, probably due to inflammation, of bystander immune responses, which at the same time is not able to control the infection, but at the same time can induce further inflammation that can be deleterious to the mice. And this is basically a working model of what we believe is happening so far. So the take-home message is that both for CD8 and CD4 T cells that go to tissues, you need expression of P2X7 for a population of these cells to survive long-term and sustain the function, the protective function against challenge responses. Of course, much needs to be done to understand the biology and the basics of that, as we saw before. So I'm going to stop here. A lot of time to thank everybody that did the work. Igor and Caio are the ones spearheading these projects. Caio also works with T-cell exhaustion. A lot of the work that we done with ZEP2 was done by alumni. Trupti as a postdoc, that now is in industry, and Sarah Van Dyke was an undergrad, that now is at UCSD as a grad student. Now we have a lot of collaborators for these projects, including Regina USP. We have to thank all the funding sources, and with that, I'll happy to take any questions if we have time. Thank you.
Hi, Henrique. Thank you very, very much for your talk. We do have time for questions, and we can open for the students first.
Sounds good.
I think we do not have questions from students here from our end, from São Paulo or from Uberlândia. Do you have any?
You guys don't be afraid. Like, you don't need to be afraid.
Henrique is friendly. You can ask.
No.
Hi, Henrique. Very nice work, beautiful work. So I have one question, a simple question. Do you believe another P2X receptor can contribute to this TRM development, or
that's an excellent question. I think they do play a role when we have evidence for that. We especially need receptors such as P2X4 for the initial activation of T cells. This has been shown by a lot of researchers, a lot of those European prunergic people, that you need expression of this high affinity receptor for ATP for the initial activation of T cells. And of course, if you need that for initial activation, you're going to need that for everything that's happening after it. We also have another research that's ongoing that right now is kind of paused because we need to hire someone to work on it on a receptor called P2X3, um, suggesting that you need P2X3 for the normal Th1 development of CD4 T cells. It's kind of a out of the box project because P2X3 is an extremely high affinity receptor for ATP. So we're still trying to understand when and where and how the heck this receptor is driving that. But it is highly likely that at least P2X4, P2X3, and I think P2X1 can be expressed by T cells could be important. Um, there's also another possibility that these receptors they have some kind of redundancy with P2X7, at least after they form these memory populations. So if you think about situations of memory, in most cases, what you're going to have is that inflammation is going to recede. So you're not necessarily going to have a lot of ATP. But I do believe that it's quite possible that the other receptors are involved. So,
Oh, thank you. Just one more question
hmm?
because I'm curious about that. So, in different tissues, you have different phenotype of T cells or different type of immune cells, initial immune cells.
Hmm.
So, do you believe that in different tissue, these T cells that enter in the tissue express different levels of these different P2X receptor or not?
Yeah, it's a good question. I mean, I don't know about the other P2X receptors. Part of the reason is that we don't have good antibodies for flow for a lot of them. Quite annoying. But for P2X7, this is actually true. We know that in certain tissues like the skin, the expression of P2X7 is low. So the effect of P2X7 for skin TRM formation, at least the CD8 TRM formation, is not that relevant. So even if you look at a transcriptional level, so that's probably related to upstream regulators of P2X7. So, for example, one upstream regulator of P2X7 that we all know is the retinoic acid. They're usually super high in places like the lung and the gut. So that's one of the reasons why, if you look at tissue resident memory cells, especially in the guts, they express so high levels of P2X7. But not only I believe, but it has been shown that the expression varies per tissue. I mean, like this is something that I mean, I had that data, but you kind of want to simplify the presentation. Otherwise, it's going to be way longer than 1 hour. But it's a very good point. And I don't know about the other P2X receptors. We eventually should look at that for P2X3 for sure. It's a good question. Maybe Hobson knows the answer. Or Regina. Regina is on the meeting. It's Regina's birthday today, by the way. I don't know if you guys know that.
So Hobson left a message thanking you and saying that it was a great talk.
Thank you so
Hobson, go
much.
to you here. Do you have more questions over there? I have a question here, Diego, but if there are other students, we can.
I think Maria Bello also raised her hand.
Yeah.
Okay, so Maria, you can ask first. I'll ask after you.
Yes, thank you. So fantastic talk.
No, thank you
Really,
so
I have
much.
so many questions. I don't know.
We'll have
We-
time to talk. Let's say.
It's wonderful work. Congratulations. So I'll begin for the simple one. Okay, so these are even the TRH cells are like memory cells because they are there for a
Yeah.
long time after, and even when the infection is over already, and
Mhm.
they still on the tissue, right?
Yeah.
So where does the ATP came from? If you don't
Oh,
have infection anymore, can
yeah. So
it
that's
be
a yeah.
microbiota?
Yeah, that's a good question. Actually, Igor, part of the research that Igor wants to do as a faculty member, so he's looking for faculty jobs. I should advertise that. I mean, it's like apply for fellowships. He wants to go back to Brazil as a faculty member. He has a part of his goal and my goal as well is to understand the sources of ATP, especially in the lung. And one of the things that we believe is that there are structural cells, epithelial cells and endothelial cells, that are part of the eyeball structure. So the TRH cells, they need to be in this eyeball structure. Eyeball is inducible bronchial alveolar lymphoid tissue, some stuff like that. So basically, it's a formation that resembles a TLS, a tertiary lymphoid structure, that is formed by B cells, CD4 T cells. You also have dendritic cells, but you also have in adjacency to those endothelial and epithelial cells because it's close to the bronchial areas. So Igor actually has measured that by immunofluorescence and by using cell lines that, of course, B cells express Pannexin-1, T cells, but also these endothelial and epithelial cells that can express Pannexin-1. So what we believe is happening is that you have an ATP donor cell in these eyeball structures that is sustaining this response. And that's very important because for the TRH formation, it's kind of a two-step formation, right? You need these cells to be able to get into the lung. And Igor actually and I, we have published that a couple years ago that in the context of flu infection, they need expression of CXCR3 that's dictated by P2X7. Once they're in the tissue, the ones that are in the eyeball area, they need to sense different signals to acquire this TFH-like phenotype. One of them is an antigen presentation by B cells, actually. But the other one now that we're seeing is extracellular ATP. And we believe that there is a donor cell via Pannexin-1 that's spitting the ATP out of the cell. Now, how and where and when this is happening, this is something that what we need to do is really go deeper on Pannexin-1 specific knockout system. So we have our Pannexin-1 Flox mouse. We want to cross that into different types of lung alveolar versus lung epithelial tissues. So that's what we believe is happening. So.
And maybe look to germ-free mice to see if the signals.
That's actually another good point. I mean, like with regards to flu, we also have to remember that the virus is eliminated, but you have ancient depots. But that's a good point. I mean, I never thought about doing that. We do know that in the gut, when we use antibiotics to remove bacteria, the amount of ATP in the lumen of the intestine decreases. And that is not only us. I mean, Fabio Grassi has shown that. So we never test that for the lung. That's actually a good point. We'll talk to Igor after that. I mean, germ-free is a little tough initially, but we can always start with antibiotics. So it's a good point. I never thought about that.
And the second question, if I can,
Sure.
is about the fact that you have more B cells in the lung of the knockouts. And so I remember that there are some papers showing that some follicular helper cells can also have cytotoxic function, even if they are CD4s. So what do you think about the idea that these resident helper cells also, do they have cytotoxic activity?
That's an excellent question. I think based on the data we have in normal, quote unquote, influenza circumstances, I don't think so. At a gene expression level, they don't seem to express a lot of things like granzyme or perforin, et cetera, et cetera. But we have another side of our research, which we're trying to understand how these cells can regulate pathogenic responses to severe infections in the lung. So typically, if you have a severe influenza infection or if you have a severe bout of COVID-19, what you have is a lot of damage, right, in the tissue. And in a lot of patients, this damage is not corrected over time. That's part of the basis of long COVID, for example. And we have actually a collaboration with GSUN at University of Virginia showing that both CD8 T cells that are resident memory and CD4 T cells that are resident memory, they are at least associated with this long-term damage formation because these responses by these cells is dictating the accumulation of fibrotic tissue, and that's partly due to cytotoxic function against alveolar cells, et cetera, et cetera. Now, where did the CD4 cytotox enter here? We did RNA-seq of these TRH cells that are present. They're actually present in higher amounts in both in mice, and then also we're looking at patients that died of ARDS. These TRH cells, they have a weird phenotype. I didn't show here because we didn't have time, So now we're starting seeing, together with this TRA signature, genes such as granzyme B, granzyme A, NKG7. So we are investigating whether you start having a cytotoxic function among these TRH cells. So I believe that in pathogenic responses, you're gonna have these cells there, and both me and Igor believe that. So that's a good point.
Thank you so much. Yeah,
No problem.
very nice. Thank you and good luck.
Yeah, well, we'll need it.
We always
Thank
need
you,
it.
Maria. So I'll follow up on Maria's question, since a couple of her questions were also mine. But I wonder if you find, Henrique, if you look at viruses that induce different levels of tissue damage, do you find also variable percentages of those cells that express the P2RX7?
Yeah, so that's a good question. We haven't done that yet in the lung. We have the X31 lab adapted strain. The one is a little less aggressive. Igor is establishing those experiments because we need to do heterologous challenge. What Igor has done is infecting mice with different doses of the lab adapted influenza. It's kind of a readout for that. Part of the reason was a grant that we submitted. The reviewer wanted us to do that, but we also were curious about that. It was a good point. And now we do see a dose response effect with the severity of infection and the presence of these TRH cells expressing P2X7. And the coolest part is that now we have access to fresh lung tissues of patients that either died of an accident, I don't know, like got shot or something like that. They're healthy controls, quote unquote, right? And patients that die of ARDS. ARDS is acute respiratory distress syndrome. And that can be related, in many cases, to flu, that can be related to SARS-CoV-2, that can be related to asthma. Anyway, in these patients that died of ARDS, so we have a low N on that, so that's why I didn't show anything. You tend to have more TRH cells, and those TRH cells express P2X7. So the severity of a lung infection is going to dictate how much you're going to have of those cells expressing P2X7, both in mice and humans. So
Okay,
I
yeah,
don't know about different types of viruses, though. That's a good question.
So the second question on that end of more applicable ends is if you're able to get samples from COVID patients, both the ones that had mild disease, stronger, more severe disease, and the ones that actually didn't survive, to see if you get level, if you have polymorphisms, if it could be caused, if it could be a cause, or if it's a downfall from the more severe tissue destruction.
So that's a good question. So that's where our collaborator Ji-Sun is going to enter. He has a connection with Peter Chen from I think City of Hope, and he has access to BAL samples, not lung. And part of the research that we're going to do together, we actually just got a fundable score in NIH grant. Fundable, assuming that we have an NIH by the end of the year. That's another story. That we're going to investigate that initially mice, but also he has the access to these samples. So we need to look. Initially, we're going to look at the expression, but this is actually something that Igor always has the curiosity to look the polymorphisms. When we did our cell reports paper a couple years ago, he did connect with some people in WAMFI. Actually, he was at WAMFI before. He was able to get these samples from patients, but we want to look at that. We haven't yet, but we do. So it was a good point.
Okay, I have a couple more questions, but I will go to Tiago first and then to São Paulo. And then if we have time, I'll ask you. If not, then I'll send the questions to you. Thank you,
Sounds
Henrique.
good. Thank you so much.
So exciting. Tiago.
If it's possible, I would like to make one question.
Sure, Robson. I didn't see your hand. So Tiago could ask a question and then go for you. That will be okay?
Mine is quick. Very nice talk, Henrique. I'm an innate immunity guy. So I was thinking regarding your talk, if you have the lack of the ATP receptor in the antigen presenting cell, what happens to memory cells? Is there an effect or no? Or
there's probably an effect. I mean, like I need to remember. I think someone did that before. I don't remember if they looked at memory cells in a very systematic way, but they certainly have a problem in the accumulation of antigen-specific T cells. We have done experiments in the past, meaning me, when I was a postdoc, where I adoptively transfer the P14 cells into their wild type into mice that are full knockouts. Not perfect, but in a way, this is more or less like understanding the extrinsic role. And you do have an issue with the formation of memory cells as well. So it is likely that you have both things being important at the same time. Because if you think about P2X7 is important for the NLRP3 inflammasome activation, which is probably going to be activated in response to viral infections. And that's going to help in the maturation of the immune system in general. So we just didn't really focus on that yet, but that is probably important for memory response.
Thank you.
Thank you, Robson.
Hi, Henrique.
Hey, Robson, how's it going? I still owe you that page.
Yeah, I have a couple of questions, but I will prepare only one. What about CD39 in TRH cells? Because in the lamina propria, at least, when we have downregulation of CD39, the T regulatory cells start to die. So in your hands, you have upregulation of P2X7 in those cells. So how is CD39 expression?
So that's another thing that we need to investigate about the survival of these cells. That could be part of the puzzle. These TRH cells, they express high levels of CD39. So it is possible that one of the reasons why these cells need P2X7 and don't die is because they have CD39. So that is another piece of the puzzle that we may have to do for this paper. Like a very simple. This is something actually we might work together with G Sun's group. They've been looking at CD39 and CD38. Mostly they're looking at CD38s, but they have the CD39 knockout mice. So it might be easier than just call Simon Robson. And because we also have the heat embargo here in Phoenix. So we have to wait until it's winter to be able to import mice. It sounds ridiculous, but it's 45 degrees Celsius here every day. So anyway, it's a long story, but it's possible that CD39 is helping these cells not die. It's not that it's promoting their survival, but it's helping them not die. And it is true, the CD39 is a marker of tissue resident memory cells. A lot of them do express CD39. So, but the other thing I wonder though, that's a part of the conversation we can have later, is that we always think about that as avoiding death because of ATP, but we also have some other preliminary studies on whether adenosine could be helping the survival of these cells. I wonder if that's part of the puzzle as well, but we'll talk about that later.
Good. Thank you.
Very good. Thank you.
Okay, so Henrique.
Hi.
Diego here from São Paulo. How are you?
Hey, Diego. How are you? Good. How about you?
Good. Everything's fine. So I was thinking about these memory cells and the role of the receptor for ATP. But in all of the studies, actually, the mice are recovering from an infection, right?
Yeah.
If you translate that to vaccines, so routes of vaccination. Have you tested regular routes of vaccination if ATP production or release is important? And on that sense, I was also thinking about adjuvants, like oil-based adjuvants, saponin-based adjuvants. Could ATP released by death of cells be an important?
That's an excellent question. And actually, Igor is doing some experiments right now. He has the adenovirus vaccine, the flu adenovirus vaccine. So initially we're testing the intramuscular route of immunization. And then what Igor is doing is that he's adding the P2X7 agonist intranasally. So we need to see what happens. I mean, I don't think the experiment is done yet. Another thing that we want to do eventually is doing the intranasal adenovirus vaccination plus the ATP agonist adjuvant. But yeah, that's one of the things that actually I was chatting with Dave Mazepust about this work, and he did suggest us to understand, to track whether this is going to be important or not. We do have another work that's Bruna is doing in our laboratory with allergy, and we're trying to look at the different routes of immunization and what the effect of P2X7 is happening on that. That's a different story, though. That's not a vaccine, right? That's allergen exposure. But we're doing these experiments right now. Hopefully by October, we're going to have that data. But that's a good question. It was we were anticipating people were going to ask us that question, so we need to do that experiment. So it's
all right. Thank you.
Thank you.
Thank you. So I think the last two questions, if nobody comes up with anything else, will be more of a curiosity. Sure. So you show very beautifully if the cells do not have the P2X7 receptor, that they localize differently, right? You have the B cells that are clustered together and the T cells that are separately. And I wonder, back in the day, I worked with, I stayed for a while in the Harvey Kantor's lab, and thinking about non-classical CD8 T cells, Q1 restricted, they're very important in viral infection. I wonder if you have looked, if they do express these receptors, if they would be present in this context specifically, and what is the phenotype of the T cells that do show up there when they are not, when the P2X is not there?
That's a good question. First, you were talking about like the NK positive T cells, like NKT cells, is that? Yeah, so NKT cells express a lot of P2X7, especially the NKT ones. We actually published a technical paper when I was still in Steve's lab. It was together with my buddy. He was a grad student at Chris Holquist's lab, and we're basically looking at recovery of cells during tissue processing when you block ARTC2.2, and you can recover a lot of NKT cells, in part because these NKT cells express a lot of P2X7. Now, I have never really looked at whether you have NKT cells in the lungs in response to flu. I mean, they are in the lungs naturally, right? At least in mice, right? I don't know. I need to look at that. The phenotype of the CD4 T cells that are still in the lung in the absence of P2X7, a lot of them are what we call these TRM1 cells. So those cells express high levels of PSGL1. They produce interferon gamma. But the thing is that the typical localization of these cells is actually excluded from these B cell areas. So this phenotype that we're seeing on the challenge, that's very similar to what we see for the cells that are not TRH cells. So these cells, what we believe is that, we meaning the field believes that these cells, they're very important to sustain an immune response that's otherwise initiated by the TRH cells. And the other thing is that these cells, they need the TRH cells to reactivate, because these cells, they need the IL-21 produced by these TRH cells. So if you don't have these TRH cells, these cells, they remain excluded from these areas. Probably those areas are reaching virus. So it doesn't matter that they're producing interferon gamma, because they're not going to be in contact with the virus-expressing cells. So that's what the phenotype of the cells is. But about NKT cells, maybe we could look at that in the humans. I mean, the only thing about humans is that, you know, that humans is kind of flipped, right? They don't have a lot of NKT cells. They tend to have more mate cells. And for that, we would need another tetramer. But that's another point. So it's a good point.
And maybe those, the QA1 restricted T cells might also play a role. They definitely change the TFH cells, and they can change B cell response on that end. But not sure. Maybe that.
So QA1. Okay, so you're not talking about NKT cells.
Yeah, the NKT cells as well. But I thought specifically.
QA1. Yeah, that's a good point. Yeah, I need to look into that. So, I mean, there are antibodies for it. We can easily put that in our panel. I mean, easily being an overstatement, because our panel is kind of large already. And Igor is not here, so
so you cannot say anything about it right now, right?
No, it doesn't matter. I mean, like, I'm gonna tell him to do. No, just kidding. It's okay. He already has 29 callers in the panel. What's one more caller? Like, keep peso pra quem tá cagado já, né? Anyway. All right. Thank you so much. That was a good question.
Thank you very much, Henrique. It's very exciting work, very beautiful data, and lots of questions. Thank you again, and thank you, Maria, again, for making the bridge here.
Just want to remember everyone that Henrique will be with us in Búzios at the Immuno 2025. So you're all invited to the Congress, and we're going to have a lot of discussion about resident memory T cells and many other things, subjects. So thank you again, Henrique. And we are waiting for everybody in Búzios.
It's going to be a pleasure.
Yeah,
so that's, I think that was
more questions. And Igor is coming too.
Igor is coming too. Actually, Igor is going to be talking about the further developments of this TRH project there. So
good. Thank
Beautiful.
you.
There'll be plenty of beer as well.
oh yeah, no, that's the what's more in life than beers, right? Anyway, all right. Thank you so
Thank
much,
you,
guys.
Henrique. Bye-bye.
Bye.
Bye, everyone.
Thank you. Bye bye.
Thank you.